Welcome to Epigenetics 2. In this lecture we will talk about DNA methylation. But before we start, let's review what is epigenetics. Epigenetics is the branch of biology which studies the causal interactions between genes and their products which bring the phenotype into being. Well, it happens through several different mechanisms. One of them is DNA methylation, but also histone modification and regulation of transcription with non-coding RNAs.
DNA methylation is one of the epigenetic modifications that regulate gene expression. Today, the standard tool for studying DNA methylation is called Bisulfite sequencing. Bisulfite sequencing allows researchers to study per-nucleotide methylation status of whole genomes. The technique does not require any prior knowledge about the genome that is studied or the sites of modified DNA and how these affect transcription. This makes Bisulfite sequencing a truly unsupervised approach to studying gene expression regulation. DNA methylation is an addition of methyl group to the cytosine on the DNA molecule. Previously, it was believed that DNA methylation blocks transcription of close-by genes. Today, our understanding of this epigenetic regulation of genes is rapidly changing. DNA methylation probably has many more functions, both in health and disease, than was previously associated with it before. Today, DNA methylation and different proteins involved in this epigenetic regulation are an active area of study. Bisulfite sequencing is a standard technique for analyzing DNA methylation. Bisulfite sequencing includes bisulfite treatment of the biological samples and library preparation for NGS sequencing. Library preparation includes adapter ligation step and PCR amplification. The library is then sequenced on the NGS machine and bioinformatics analysis is performed to find differentially methylated regions in conditions under study. Bisulfite treatment of the DNA molecule changes an unmethylated cytosine into uracil, while methylated cytosines remain intact. After PCR amplification, uracil is further modified into thymine. Two different approaches to adapter ligation step of library preparation exist. The T-Bioinfo platform can be used to analyze both types of data. However, we will focus here on the non-directional library preparation technique as it is a prevailing one due to its lower costs. For directional library, adapters are designed so that only original positive and original negative strands of DNA are sequenced. In this type of library preparation, the final NGS reads will have unmethylated cytosine converted to thymines, while the methylated cytosines will remain intact. For example, in this short genomic region, all cytosines were converted to thymines after bisulfite treatment and PCR amplification. In the non-directional approach, the adapters are not specific. Thus, after PCR amplification, 4 kinds of DNA fragments are sequenced. Original positive, where all unmethylated cytosines are converted to thymines, and original negative, where again all unmethylated cytosines are converted to thymines. In addition, the library will include complementary DNA to the converted positive strand, as well as complementary DNA to the converted negative strand. When aligning non-directional library to reference genome, the algorithm will allow mismatches of C to T in positive strand, so that only original positive fragments will be analyzed, and for negative strand, G to A mismatches will be allowed. Allowing mismatches in alignment will impact the downstream calculations of per position methylation status and thus is an important step in bisulfite data analysis. Basic bisulfite workflow of data analysis includes the following steps. uploading raw genomic reads, cleaning these reads from adapters and PCR duplicates, aligning the reads to the reference genome, allowing mismatches according to the method used for library preparation, per position methylation calling, and finally identifying methylated islands and annotating these as known genes, promoters, CpG islands, or other regulatory genomic sequences. T-Bioinfo platform has Bisulfite sequencing section dedicated to analysis of DNA methylation data. The algorithms in Bisulfite section are arranged according to the basic analysis steps: pre-processing, alignment, per position methylation calling, segmentation of genome to identify methylated islands, and island annotation. Two options of data analysis are possible: analysis of each sample separately, and alternatively, analysis of pair of samples that should be compared according to the logic of the experiment. When analyzing each sample separately, one does not need to determine groups of samples. All samples can be dragged into one group, and contrast for this type of analysis will be 1, 1. As a result of the pipeline, one can expect separate tables for each uploaded sample with identified methylated genomic regions for positive and negative strands separately. In contrast, when differential methylation of two samples is required, the samples need to be uploaded into separate groups, and contrasts must be set to -1, 1, 1, -1. The contrasts ensure that both hypermethylated regions and hypomethylated regions are found. The basic pipeline for analysis of Bisulfite sequencing data on T-Bioinfo platform looks like this. First, the cleaning and pre-processing of reads is performed. Then the alignment of reads. Alignment algorithm Segmentation implements a matching strategy based on enhanced suffix arrays. The parameters for identifying library preparation method and possible mismatches between reads and reference genome are required. Per position methylation calling per nucle assigns scores estimated by Poisson likelihood function to all cytosine positions. Parameters are needed for printing result files. BNS algorithm identified regions enriched by high-score methylated positions. Finally, annotation models allow to cross BNS find segments with known annotated regions of the genome based on the GTF file. Annotated regions as found in GTF file can be extended so that BNS found regions close to the known regulatory element can be identified with this element. Additionally, more stringent BNS region filtering is possible. Alignment of reads in both options of analysis, no contrasts and contrasts, will be similar and will be performed on each sample separately. In non-directional library preparation options such as Methyl-C, two types of mismatches will be allowed when aligning reads to the reference genome: C to T mismatch and A to G mismatch. When reads will be aligned, the algorithm determines which strand the read belongs to. Positive, no special manipulation of read was needed for the alignment. Or negative, read needed to be reversed and complement needed to be aligned. Thus, after alignment stage of the pipeline, two result files are expected per sample: positive and negative strand alignments. Per position methylation calling will be performed for each SAM file separately in case of no contrasts, or using contrasts on negative and positive SAM files separately. When no contrasts are needed, for positive strand, all cytosine positions will be analyzed against thymine, and statistics will be calculated. The scores are based on the rate of methylation estimated by Poisson likelihood function. For a negative strand, all adenine nucleotides will be analyzed against guanine and statistics will be calculated. If contrasts are chosen, the subtraction of scores calculated for each sample will be performed. The resulting score will be given. Due to the impossibility of segmentation of genome based on negative scores, two contrasts were selected. This ensures that both methylation status when sample 1 is more methylated and when sample 1 is less methylated than sample 2 will be identified. The per-position methylation results can then be analyzed for methylation islands when speaking about each sample separately, or differentially methylated regions when taking contrasts into account. These found regions of the genome can be annotated according to the known genomic regions, as indicated in the GTF file. Expected output offers annotation model will be tables of crosses of the found methylated regions with chosen regulatory elements. For each contrast and each strand, please find separate tables attached. The table is divided into two parts by a delimiter. The first part contains the known annotated region, and the second part the found methylated region with start, end positions, and segment score and average. Result files of the Bisulfite-seq pipeline can be found in the Download folder. This folder contains two types of results: BINS segmentation result files for each strand separately and depending on the contrast option, separate for each sample or for each contrast. And additional type of result files are the zip compressed files containing annotation results. When extracting these files, several tables will be available for each contrast. The most full table in each folder containing most information will be of kind promoter intersect table. Each strand and each contrast will have a separate table. As a final step of analysis, all results should be replicable. If analysis was performed on each sample separately, we recommend to retain methylation islands only if it was found in at least 2 samples in the group. If the analysis was performed on contrast, the differentially methylated regions should be found in at least 2 pairs of contrasting samples.